Okay, welcome everyone to Proteins and Protein Folding 5. Today we're going to continue our journey through the crazy world of proteins. And as always, we're going to start by doing a short review of what we did last time. Okay. So if you remember, if you were here, last time we talked about motorproteins. Motorproteins, basically like the name says, they're basically like a motor that they turn energy into movement, right? For a motor that would be gasoline, and through combustion this energy gets turned into movement and the car can drive. For a motor protein, this energy comes from ATP, and for instance muscles then use this ATP to convert chemical energy into movement. We were looking in particularly in detail at myosin. We had the motor subunit of myosin in PyMol, and we were looking at the movement between the different states and how ATP binding and hydrolysis basically facilitates the power stroke and thereby the contraction of the muscle fiber. Alright. Okay, so just so you remember, you had the myosin on like a bundle-like structure in between actin filaments, and upon doing this power stroke, these actin filaments moved closer together, thereby facilitating the muscle cell to become shorter and contract and therefore move. Okay. At the end of last time, we also talked about another protein called kinesin. Kinesin was the protein, if you remember from last time, which was able to walk along microtubules and thereby transport different cargo along basically like highways from inside the cell to the outside or to the outer membrane. So today we're going to look at a different aspect of motorproteins, which is motion, right? So last time we talked about muscles and how we use them and how myosin makes it possible for us to actually move per se. Today we're going to look a little bit at the microcosmos. For instance, I don't know, I hope most of you know what this is from biology. This is paramecium. And this is basically a single-celled organism which is able to move and hunt for food. It also has, I believe, chloroplasts. I'm not sure. Maybe some of them do, some of them don't. And today we're going to actually look at how this microorganism is able to move. And this has a lot to do with what we talked about last time as well. If you look in detail, maybe you can see it. Maybe it's a little hard to see in the presentation. But the main part of the movement is basically a stroke of little hairs on the outside of this single-celled organism, right? In red on the bottom, you can see this a little bit magnified. So basically a back and forth movement of hair-like structures on the cell, right? And we're also going to look today at also a similar type of movement by, for instance, or movement of sperm cells, right? Those kind of swimming motions, a little bit looks like snake, if you want to think about it like that. So very, very interesting stuff. A very, very basic mechanism that is found in many, many living organisms and cells. And probably not today. Today we're just going to get a little bit into the details of the structural basis, but probably next time we will also going to look at a different type of motion, which mainly is used by bacteria, which is called flagellar motor. Okay. So a little bit to talk about the two different words that are important today, we're going to talk about flagella and cilia. So cilia are basically the little hairs that you saw on the paramecium that are doing the back and forth movement. And the flagellum is a little bit a longer structure, and there's usually only one of it, or at least a fewer number of them on a single cell. Right? So there are some structural and functional differences, and I think at the end of this or next workshop you will be able to know what exactly the functional difference is between those two. But for now, just take it into account. Cilia are the little hair-like structures that surround the cell, and flagella are more what we saw on the last slide, or in the last here in the background. Longer structures that are fewer in number on cells. Okay, so coming back to this picture, last time we talked about kinesin. Today we're going to talk about dynein. Right? So last time we only learned about dynein because basically what it does, it transports things in the opposite direction of kinesin. Right, so kinesin goes from minus end to plus end, and dynein goes from plus end to minus end on a microtubule. Okay, today we're going to learn a little bit more about dynein, structurally and functionally, and also different areas where it appears and is very important. All right, so last time we talked about kinesin. Kinesin basically does its motion in a hand-over-hand type movement, as it would be described. Right? We talked a little bit about the ATP binding, then swinging around, binding again, and then the other side is released and swings around and binds again, and so forth, which makes it look like it's basically walking on the microtubules. So today we're going to look at dynein, and actually with you together, I want to basically discover and ask you what you think the function is or how it works, right? So we're going to use the structure, and I want you to basically investigate how the structure could lead to dynein's function. Okay. And for this, I know maybe not so many people are using PyMol today, but still I think we can do it together. Today we're going to use the protein which can be found under the archive number 5NVU. All right, so if we go to PyMol, today we're going to start just with PyMol open. No previous file is needed or anything, and we're just going to type fetch 5NVU. All right. Okay. What that does, it will get the file from the server, which is basically the file of dynein, right? So the protein structure of dynein. So right now, again, it doesn't look like much, and we have to work a little bit with it to make it more visually appealing and a little bit to make more sense out of it. Right? So again, for those you haven't read chat, maybe I would like to ask you, are you using PyMol? If yes, then type yes in the chat. I know Pavel does. Is there somebody else? Okay, while waiting for your answer, I'm going to start already a little bit working with the protein as itself. So a little bit similar to last time when we worked with myosin, here you can actually see this is also a lot of stuff that is extra that we don't need. Because if you think about, well, if you go back to the presentation, we want to have something that looks like the left part, the denine, right? We want only a part that looks like this. So, and to get this, we only want to use chain A. Right? So, for using chain A, we can simply type in the command, select chain A. Okay. And basically, this is all we need. So we go and select it. We see that it's highlighted because every atom gets like a pink point. And we can choose it here as selection. We can give it a new name. So I'm just going to call it denine. All right. Well, actually, it's not completely true. Like the whole denine protein complex is, of course, also includes this other stuff, right? And the connection to whatever cargo there is. But what is interesting for us right now only is the functional part, right? The one that does the movement. Okay, after you give it a name, you have to press enter, and then it changes here to denine. And all we want to do now is hide the rest. So first we're going to hide, we're going to press on all, then the H, and hide everything. So now everything is hidden, but we only want the one that's selected. So we click on that, and we click on S for show. And, oh no, not surface, that was a mistake. That's going to take a little bit, because the surface is somewhat difficult in calculation. So actually, so this is looks somewhat similar to what we just saw in the presentation, the kind of structure that I showed you on the other side of kinesin. But we don't want the surface, so we're going to hide it. Then we're going to show cartoon, right? Okay, and since the rotation is a little bit off center, we're also going to recenter it by just clicking on action and center. So now we can just move it around. Okay, so now everything has one color. Something that is usually very, very useful is to give everything a color, and in this case, according to spectrum. So it will start from the N terminus of the protein and gives it, I think it starts with red, and then at the C terminus it will be blue. Right, so like a rainbow. Okay. All right, and this is what it looks like. This is what we want to work with today. Right. And first of all, something that is very, very helpful to understand the function of a protein is to make a schematic representation of it. And this is how we're going to use the whiteboard today, because what I would like you to do is take a look at this and try to draw a representation of it, right? Just a very simple representation of its units and what you think can be regarded as different parts of this protein. Right? To give you an example, going to make this a little bit bigger. Maybe move it like this. And then I'm going to go over to the whiteboard. I'm also going to show you, maybe not. Yes, I'm going to show you PyMol in the corner, in the bottom corner. Maybe a little bit lower. Okay. So, and you know from the previous times, if you use the whiteboard, if you joined it, what it would like you to do is choose a color or just click on the pen and then write your name. Okay, so I will be using black. So I'm just going to write my name. So I would ask you to do the same. So I know there's a delay, so I'm going to give you a little bit of time. Okay, oh, Pavel is very quick today. Seems to have good connection. Okay, Kate, very nice. Somebody else going to draw? And don't be shy. Oh, I see, very good. There's another one. Okay. So to give you an idea what I mean, I'm just going to try to draw two parts that are very obvious, right? And so I'm going to start with the things that I think will be functional different parts in the protein. Right. So I'm going to draw this, and then I'm going to draw a long line like this. Right. Okay, so if you look at the picture in PyMol, you probably know what it is. This is the yellow part. So I think what will be functionally different is one, I would call this the foot part, and I would call this the leg part. Okay, so what I want you to do is do the same, right? So you can, of course, copy what I did. If you have a different idea of what's a different unit in the protein, that's okay. But try to find out what you think are different parts, functional parts of the denine protein, right? So try to make a schematic drawing of it. So what we want to have in the end is a cartoon representation of this whole protein, what you see now in the bottom right corner. Okay, very nice. Right. If you want, I can also, in the meantime, give you some different angles that might help. Right. Maybe from the other side. I'm going to go back to the PyMol to make it better visible for the people who don't have PyMol today. Right, so we're just gonna look at it a little bit from all kinds of angles, so you can maybe take a good guess in what's happening with this protein. Okay, let me go back really quick and see what's happening on the whiteboard. Okay, very nice. Very, very nice. Christina, try a little bit harder. I'm kidding. I know it's difficult to draw. Maybe, I mean, I have a tablet here, but maybe with a mouse or with a touchpad, it's not that easy. So even if you have an idea what its function is, you can try to give it a name like I did, right? So foot and leg. Pavel, no idea? Okay. Looks very, very nice. Let's see. Let's show everybody what they draw. Grabby thingy. Okay, good. And a donut. Okay. Those are some very, very good ideas. So you definitely can see 3 main structures. All right, if I go back to PyMol. All right. So you can definitely see 3 main structures, right? So one is the foot part that I drew. Then you have a circular shape, donut or body part. And you also have a distinctive linker part that is kind of sticking out, right? And also Aya saw very well that this body part is actually 6 repeating units, right? They all look kind of the same. And they're like 1, 2, 3, 4, 5, 6, right? In a circle. Very well. So let's see what is going on really, right? So going back. Okay. Good job everyone. That looks very, very similar to what a certain research group found, right? So their idea of the protein was basically, well, instead of foot, they called it microtubule binding. They called it not a leg, they called it a stalk. Then they have the head part, or what we call the body part, with 6 different units. They have a linker part, right, which is the one that sticks out. And actually the neck and the tail we didn't see in our model. So very well done. Okay. So, and important to know is, because we also don't have the model for it, the microtubule is of course at the foot end. Okay. So yeah, the different parts of the body part are called AAA rings. Right? They have a different number, so AAA1, AAA or AAA2, AAA3, and so on. Okay, so now how do you think this thing will move? Right? So what do you think is something that would make sense in a way that would make this whole thing be able to carry something on its tail end? So this is actually not so easy, and I want to show you something that a research group did to figure out the mechanism, right? So what they did is they took pictures of it. Of course, if you think about taking pictures of atoms or molecules, that's very, very difficult. And there's a special technique to do that, which gives you only very blurry and very rough pictures of what the protein looks like. Right? In this case, those are cryo-EM pictures. And you can see they have basically this bigger structure, and they cut like different planes through it to take a look at what the protein looks like in different conformations. Because if you think in this structure there are many dynein molecules or dynein proteins in a row, and of course not all of them are going to be in the same conformation. Some of them are going to be in one, and some are going to be in the other. So they looked at the bulk of them, or many different ones of them, and they analyzed the pictures that you can see here. And then they used the model that we just looked at and tried to think about how can you fit this model into this picture, basically, in these dark areas. And then look at how it moves to kind of show the proof of what the idea is of how this movement is facilitated. I'm going to talk a bit about it a little bit in the coming slides, but this is a cryo-EM picture of an axoneme. Basically, what is inside of a flagellum or a cilia, right? So the structure of it. And there are two rows of these dynein proteins. One are called ODA, outer dynein, maybe assembly. I forgot. And IDA is inner, right? So you have an inner row of these dynein molecules and an outer row. So they all try to line them up with a picture so they can kind of fit the molecule and then make sense of it all. Okay. So, I mean, it's probably not very obvious for people looking at it right now, but you can already see one difference, right? Between what you see here on the right, what they fitted, and the one we just looked at. Let me actually show it to you in PyMol. If I just bring the same molecule in the conformation so you can kind of compare it. Okay. Okay. All right. So basically this is the conformation of the protein in the picture from the paper, right? So let's go back. So if you look at the presentation, for them here it's in purple. For us, it's in blue, right? So big difference between those two models, the one that they try to fit and ours, is actually this linker part of this protein. Right. So, and after analyzing a lot of pictures and a lot of thinking probably, they came to this solution. Right? So, again, this dynein only functions with the use of ATP, like we talked about myosin and we talked about in kinesin. Dynein also has to use ATP to do this conformational change, to do the walking, right? So in this case, the main thing that is changing in conformation is the linker part. Right. So everything else is actually not moving that much. So you can see here, they say if ATP binds, what will happen is the foot doesn't bind to the microtubule anymore. It lifts up and it stretches out. The stretching out is facilitated by the linker basically sliding from the yellow part onto the light blue part. Right? So this is like reaching forward with the foot. Then, when ATP is hydrolyzed and is split into ADP and P, the foot rebinds at a further point as before, right? It started in the red location and now it's in the green location. And then basically what they call the power stroke happens. If phosphate goes out, a conformational transition happens where the linker slides back from the light blue unit to the yellow unit. All right. So maybe a little bit to imagine the transformational change. We don't have a fitting structure today, but something that I wanted to highlight is, actually I'm going to show the surface again for that. If you look at the surface, right, the part where ATP is binding is actually here in this top part, very close to the linker. Right, so it goes into this, you can almost say it's like a pocket. And it changes, it's kind of I'm not sure about what exactly happens, but if it binds, it expands this pocket or it makes it tighter. And therefore it changes how this light blue unit interacts with the linker. Probably it will loosen its grip and the linker can then slide down towards what we can see as a, well, yellow-greenish area. Right? So then if it goes out, this again wants to bind in this area more than in the yellowish-green part, and then moves back. Okay. For this, they also had a animation. So in the background here, you can see actually the cryo-EM pictures, which is very interesting. Right, so only what you see is those like shape-changing black rings basically, and maybe part of the foot in the back. But if you have a lot of them, you're actually able to fit the model with quite good accuracy into this kind of shadow. Right? And they can confirm how this transformational change is then also moving the whole row of dineins to the left. Plus minus, right? Opposite of kinesin. So we're going to talk about how this turns into movement in a second. What that means for instance, transport along microtubules in the cell, which we saw with like kinesin basically doing the thing in the opposite direction. Now what we can see is adenosine uses a very different mechanism from kinesin to walk in the opposite direction, towards the inside, towards the nucleus of the cell. Alright, so this is almost like actual legs, doing like a left-right, left-right stepping motion. Right? Okay. So this protein is not only essential for the transport of vesicles and organelles in the cell, but you can actually also find it in what I talked about, in the cilia, in the flagella. Right. So already, if you look at the cilia and flagella under a very good microscope, and you cut through them, what you will see is structures like this. Right? You will see basically 9 double circles in a circle and 2 round circles in the middle. And this has a special name. This is called an axoneme. I hope the pronunciation is correct, but I think it's called an axoneme. So this axoneme has a lot to do with how movement for cells works. Right? For single cell organisms, bacteria, and not only movement. We're also going to look a little bit at other functions. Okay, so going a little bit more into the details of the structure, you can see the axoneme basically starts from the centriole. I'm not sure if you remember that from biology, but from inside the cell is then built towards the outside to form this kind of hair-like structure. And in a cut through it, we already talked about the ODA, the outer dynein, and the IDA, the inner dynein, which we saw in the picture before. And basically the green parts are microtubules. These are not like the microtubules inside the cell. Those are actually linked together by other proteins called nexine. And they're also proteins that act like a spoke in a bicycle. These spokes also are there for structural integrity of the whole axoneme. Right. So, and in the center you have a central bridge and singlet microtubules, and the outer ones, they're more like double circles, and they have a subfiber A and a subfiber B. Okay, so let's look at how this, if you think, how can this transport of, or this function of dynein, of transporting, for instance, vesicles and organelles through the cell, how would it be translatable into movement of a hair-like structure like this, of a filamental structure like this? It's actually very, very interesting. Right? And actually the same group did this analysis and also found out how basically not only the movement, right, what they theorized, what it looks like, but they also found out how this movement leads to a movement of a flagellum or a cilium. Because what we looked at is basically in the pictures before, we looked at the cryo-EM cuts of dynein from the side and the different conformational changes, but they also analyzed what does, in what part of the flagellum or cilia does the dynein have what kind of conformation. And what they found is very, very interesting, because what you can see here as blue dots and red dots and green dots are basically different conformations of the dynein between the microtubules, right? So only in one area are microtubules basically in the power stroke formation, and the other one they are in the relaxed formation, or the relaxed conformation. right? So if you think about it, on this part, on the outer part, like this is like only a short part of a flagellum. On the outer part, with the red dots, those are all doing the walking motion. So they're basically trying to move along the microtubuli. Of course, there's a limit, and they're basically stressing the whole structure and making it stretch on the left side of the flagellum. Whereas on the other side, they are all in the relaxed state, so there's no stress happening. So similar to, what's a good thing that's similar? If you think about it, for instance, if you would have a metal pipe and you would only heat it on one side, right, only on one half, what would happen is the metal would expand, stretch out, and actually cause the other side to, the other side that is cold, to then bend away towards it, towards the cold side, basically. Okay, so here's a symbolic representation of that. Right, so the right side, so you have basically blue, a microtubule, and here another microtubule. And in the middle, the red is a denine. So basically, if the left part makes a power stroke, this will cause the whole structure to swing to one side. Right? So you basically have a switch between this part and this part. Okay. And there's also a very good picture which kind of summarizes this whole action. And actually today I don't want to overload you with a lot of information. So actually this is all that I have for today. So this kind of summarizes what we're talking about, and I'm going to show you a little bit more after this, because maybe you think, yeah, well, this motion, well, how does it apply to our life at all, right? So why do we need to know about cilia? Okay, but first, take a look at the video. Oops. Whoop. Okay. Let's see. Okay, give me a second. I guess I have to do a window capture or display capture. Okay. That's okay. I can very quickly watch this video just on YouTube. Okay, I also need to turn on the sound. Okay, there we go.
Motile cilia and flagella are hair-like appendages of cells that generate fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualized the molecular machines inside the flagella through cryo-electron tomography. Thousands of motorproteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, The cargo is pulled forward by the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However, if dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism generates the flagellar beat. As one side is inhibited, the other side is active. In our model, regulatory signals result in inhibited dyneins on one side. This allows dyneins on the opposite side of the flagellum to walk along the microtubules. Molecular linkers resist this sliding, which causes the flagellum to bend in one direction. By periodically switching the side of dynein inhibition, the flagellum alternates the direction of bending, resulting in the typical whip-like motion. The propagation of dynein inhibition in different regions of the flagellar wave along the length of the flagellum leads to the characteristic beating motion. The dynein inhibition may occur in an all-or-none fashion, similar to an action potential, which could explain how cilia and flagella can switch their bending direction back and forth in less than 1/100th of a second and continue beating for days.
Okay. Very good. So, this is basically how flagella and cilia work. Just as a note, it might happen that today at 6 o'clock exactly, my internet is going to go down. I actually totally forgot about it. So probably next time we have to think of a different time when to do the workshop. Because, I don't know. Don't ask me why this happens. It's just something that my internet service provider decided to do. So the stream might go down. We're going to continue a little bit afterwards. Okay, so where do we find this in actually humans? Right. So contrary to what you might think, in humans this type of movement is actually also very, very common. One of the most important areas where this happens is, for instance, in the lungs. Here is a short, just a very, very short video of what is happening in your lungs actually. Right, okay, one more time, this is very, very short. Right. So those are cells on the inside of your lungs, which are lining the bronchia and also the alveoli. Right. So as you can see, these types of cells possess cilia that are moving regularly in a wave-like fashion. Test. Okay. Uh-oh. Okay. So apparently, restarting streams automatically mutes you and the chat doesn't work anymore. Um, okay. I guess, I mean, we finished the main part talking about Celia and how flagella worked. I think the last part that I talked about, I will leave for next time because maybe, I don't know, some people dropped off when when the stream disconnected, or maybe, I don't know, just so to make sure that we go over it again. Um, Not seeing the chat though. It's a real problem. Okay. Okay. Now we see actually what people wrote in the chat. It didn't update for me. Um, okay. To About the part that you've heard, are there any questions from the people that attended? Let's see. Oof. That's a tough question, Pavel. I don't know exactly. Um, I would have to look at the yeah, I would have to look at the publication. That's not a number I saw or that I could maybe um, judge or estimate. Definitely sounds like a lot of ATP is required. Okay, any other questions? Okay, if there are no other questions, if you're interested in the topic or maybe get a question anytime, any other time, standing on in the shower or whenever, you can always send me a message and I'll try to answer whatever questions you have. Okay, then say thanks for coming today. And next time we're going to talk about what you see. I think you didn't hear me talk about it, but this is basically like a propeller in a U-boat or well, in normal ship as well. But this is what bacteria use to um, move through a medium. And this is what we're going to look at next time. Okay, see you then. Thanks for coming. Bye bye.